scientists have announced they found a new way to cure diseases. When a disease is caused by a bacteria, instead of just killing that bacteria, maybe instead we should be adding more bacteria to cure the disease. And so I've come to this lab in Oxford to speak to the lead scientist on that paper. She is also a fashion designer, a seamstress. She's a model who models in Vogue. She's a great friend and my biology partner in crime. It's Suzie Ford.
Hey.
Hello.
You're right.
I am.
Good.
We've been at Oxford together a long time, haven't we?
Some might say too long.
Too long. 6.5 years?
Yeah, I think 6.5.
We did our undergrad together, we did our PhDs together.
Yep.
So you got a new paper out.
Yeah, I'm really excited.
Yeah, you're the lead author.
Oh my gosh, don't. It sounds
proper
weird.
Published scientist in Nature Communications.
I know, right?
Super high profile journal.
I basically peaked now.
You have?
Yeah, I mean, it's going to be down.
Downhill from here.
Absolutely downhill from here. I'll just go, so, you know.
What's the title of your paper?
It's called Microbe-mediated host protection drives the evolution of reduced pathogen virulence.
Otherwise known as good bacteria make nasty bacteria less nasty.
Exactly.
And
I should have.
What we're going to be talking about.
I should have just called it that.
So yeah, tell me a little bit about what you did.
So basically I worked with this little microscopic worm. It's like 1 millimeter long, and I infected it with stuff, good things and bad things. And I saw how the bad things changed. So I use the C. elegans, the little worm, as a model host.
Caenorhabditis elegans.
Caenorhabditis elegans.
Caenorhabditis.
Yeah, exactly.
Yeah.
So I use that as like a model host organism. They're a really, really good model system. So not only are they transparent, so you actually can see the infection, and they're kind of tiny and manageable. You can have loads of them and they replicate really quickly. So you can do evolution experiments quite quickly.
So we're talking about good and bad bacteria. What are these bacteria?
So we chose Staphylococcus aureus, which is, the big sort of superbug.
That's the SA in MRSA.
Yeah, the SA in MRSA.
MRSA, as the Americans call
Yeah,
it.
well, yeah. So basically we used one that wasn't resistant to antibiotics because that would have been a little risky. Yeah. So Enterococcus faecalis is the good bacteria that we used. Basically a gut microbe.
Okay.
And we get it in our poo because we poo our gut. Okay.
So did you have to go through a load of human poo to get this bacteria?
No, we didn't. So this one's well studied and we actually just got it from a friend, so it's fine. A lab friend.
Okay.
Like they had it in their freezer, it's fine.
How do you go about infecting a worm?
The system's really cool. Basically, the worms eat the bacteria as a living. So, it's called bacteriivores. Like we're herbivore
omnivores,
or omnivores.
carnivores.
Yeah, we're omnivores or carnivores or whatever you like to call it. They are bacteriivores. They eat bacteria for a living. So you basically put a culture of bacteria on a petri dish. You let it grow overnight. So there's a nice lawn. We call it lawn. I don't know why. It's
you
weird.
had to mow your bacteria.
No, you don't. They're good. The worms do it for you. Yeah. And then you put them on, and they just munch away to their death. Unless, of course, they've got the good bacteria, which is great. Yeah, it's fine.
And then you said that you had to get the bacteria out of the dead worms that died.
So this took a lot of pestling, like literally pestle, mortar, crush. So this was the bane of my life. I don't know if you can see, but these were the nice little pestles that I used to crush apart the worms to get all their gut bacteria. A lot of blisters from these guys, not my best friends.
So you're picking up these microscopic worms.
With platinum wire, it's like 1 millimeter thin, under a microscope. It takes a lot of fine motor skills.
Yeah. And then you're squashing them down.
Yeah, it's kind of gross.
Trying not to get any other atmospheric bacteria.
Yeah, so we do it in this sort of safety cabinet. This is where I sat and crushed up worms, and because that was so brain numbing, I used to watch films and stuff, so I set up my computer here. I probably shouldn't be telling you this,
What's
but
the best film to watch whilst?
I actually because I needed hours and hours and hours worth. I just watched there were 6 series of Pretty Little Liars, and I just it's so awful. But I would just watch hours of Pretty Little Liars just with my headphones on and just crushing constantly. It was a nightmare.
So you've got kind of dead worm paste.
Yeah.
What do you do with your dead worm paste? How do you get the bacteria out?
For the evolution experiment, we streaked it with a loop onto a petri dish and let that plate grow overnight. And you wake up to a nice plate with loads of colonies grown all over it. And then you pick the ones that you like best. Well, you don't do that, that's bad science. You just pick randomly. Yeah.
And then you spread them onto its own new bacteria lawn.
Yeah, you let them grow, and then you get the next generation of worms to eat that and do it all over again.
So you've allowed these nasty and nice bacteria to evolve.
Yeah.
And start infecting worms together. Did the bad bacteria change after all of this?
Yeah, absolutely. So we found that it became way less harmful to the little worms. So it could either become really harmful and do really well in the worm, or it would have to resist the defensive microbe. Not only is the defensive microbe killing the pathogen, which is great for us, but it was also stealing the pathogen's most useful proteins that it was using to get energy. Siderophores are basically proteins that any microbe produces, and they basically bind iron in the environment.
And so bacteria need iron to grow, and then they mass produce these proteins to collect in the iron.
Yeah.
But then some sneaky bacteria come along and
steal them. Yeah. So that's what we found was actually the mechanism that was going on. So it was kind of like using two weapons. So killing and stealing. So in response to this stealing and killing, the pathogen evolved to produce fewer siderophores to stop the defensive microbe from growing so well.
So it's starving out the good bacteria.
Trying to starve out the good bacteria, but at the same time, it's kind of starving itself out as well. So although that's really beneficial when the defensive microbe's around, when the defensive microbe isn't around, it's just going to grow really poorly. There's no benefit of it at all. So that's why it's just less harmful in the absence of the defensive microbe.
I hope you enjoyed this with Suzie, who has been pestering me for the last 6 years to be in one of these videos. Thank you very much, and bye!
Bye!